Hello and welcome to this week's What Were They Thinking? This week we have a variety of news items, although strangely very little on the CCP virus. Instead, plenty of other new and exciting tropical diseases. The similarities between your brain and a pair of testicles, and what China's artificial sun is doing. Timestamps for each of these can be found below. Let's start with new diseases. After all, they've been bringing everyone joy and happiness for the last year or so. In this case, the H10N3 bird flu. This is effectively the next major outbreak in the making. The last time we had a major outbreak of bird flu was 2005, and at that time, it spread very quickly and led to a much higher incidence of death than normal influenza. The concern with the CCP virus was that we might see something very similar, a fast spread, long duration in the population, and a high death toll. Fortunately, that's not what we're seeing. The similarities to the bird flu, however, are somewhat more. Particularly, this outbreak has also started in China. Fortunately, that's where the similarities between the CCP virus and bird flu end. Although one patient has been clearly identified and no others have, there is a constant reservoir for bird flu amongst birds in the wild, and on occasion this gets into chickens and other domesticated poultry. While still on the topic of influenza, we have an interesting article on the way 2 human influenza virus strains went extinct. Consider just how difficult it is to control influenza at the moment. Every year we need a new vaccine, and that vaccine takes a long time to develop. In doing so, we can barely control the disease, let alone stop it in its entirety, and yet at least 2 examples have gone extinct. These 2 examples have disappeared over the same period in which the CCP virus has been most active. This can be attributed to a number of things: wearing masks, greater awareness of hygiene and dealing with things like viruses, the isolation policies that were brought in various countries, and a few others. All of these have led to significantly lower than ever before rates of influenza, and this is why at least 2 of the types that have been a problem over the last few years where we've kept records are now gone. Of the 2 subtypes that are currently circulating, there are certain clades, and clades are a further breakdown of the virus types that have disappeared. One is the B Yamagata, and the other is the 3C3A. Neither has been detected since March of 2020. This will, in theory, make it much easier to develop vaccines. We mentioned early on that you need a long lead-up time, and every year you need to change the vaccine. This means that it takes a lot of effort and resources. Before you even get to the point where you're making a vaccine, you need to choose which vaccines you're going to develop, and specifically for which influenza viruses. Having removed 2 clades, they're now able to have a much smaller pool of viruses that they can target and develop a vaccine for. This means rather than choosing 4 or sometimes 3 of the viruses out of 10, you may now only have to choose 3 or 4 out of 8. This gives much better coverage. Going now to a very different disease and a trial of what happens. We're looking at malaria. Malaria is a parasite, which compares somewhat more unfavorably to a virus. Parasites need to move between hosts and generally require a vector, in this case mosquitoes. Something like a virus can be spread from host to host almost directly. Parasitic diseases like malaria are what gave rise to treatments like quinine. Our understanding of what malaria involves, though, is somewhat limited. In fact, treatments for malaria are delayed because we don't quite understand how it works and how the parasite gets into its host. This is where a number of volunteers come into play. They volunteered to be infected by malaria. The study is limited, and for understandable reasons. 7 individuals are who volunteered, and 7 is a fairly large number for injecting them with a parasitic organism, and then waiting to see just which organs are affected first. The theory being that the spleen would be targeted first because it's full of young red blood cells that are one of the preferred hosts of the malaria parasite. This appears to be supported by scanning of the spleen, which showed much higher glucose use. And, among other things, it seems to be much greater activity, which could be a combination of either an immunological response and or the parasites. Exactly how isn't clear yet. What is clear is our next study. It's well known that men tend to think more with what's between their legs than what's between their ears. And this is perhaps for good reason. In terms of evolution, this makes sense. The more reproductively successful a male is, the more successful they are overall. Biomedical scientists from the University of Aveiro in Portugal were looking at the similarity in proteins between testes and the brain. What they found was that there was a very large number of common proteins. In some cases, it could be argued that the male reproductive system does most of the thinking for the males involved. In other cases, some have the ability to temper their testes with some sort of cognitive function. This study was fairly comprehensive and looked at 33 tissue types, things like the heart, intestines, cervix, ovaries, placenta, testes, brain, and more. The study found that there were about 13,000 proteins in common across this set, and many of them had a higher degree of correlation in the brain and the testes. The underlying theory for this is that the testes have to produce sperm, and the role they have there is energy intensive and has some very similar underlying physiological processes. These are similar to how the brain operates, and specifically the neurons within it, which are also very energy intensive and also need to have a number of physiological processes for which parallels exist in the testes, particularly the movement of content within the neuron to outside the neuron. While still in the world of genetics, but this time looking at chromosomes, scientists have been able to weigh the mass of a chromosome. Chromosomes are the very, very small parts of your DNA. They're like small sections that can be neatly packaged up. The human body normally has 46. 44 of these are related to somatic function, that is, your body's general needs. 2 of these are related to sex. These are your X and Y chromosomes in the case of a male, or X and X chromosomes in the case of a female. Understand that getting the amount of DNA in a cell as a whole is difficult, but measuring a fractional amount of this from a chromosome and being certain that you've got the whole chromosome's weight is much more difficult than you would think. In fact, it's so difficult it's taken us a very long time to understand how to do it and do it effectively. It involved the use of an X-ray device, a very powerful X-ray device. They needed to use it because the amount of material you're actually going to measure in each cell is a fraction amount of anything. In fact, 242 picograms. A picogram is like a regular gram, except you need to put 10 zeros in front of that value. For those of you who insist upon using the imperial system, that's 8.5 ounces to the negative 12th power. Now you understand why it was so hard to measure an individual chromosome. What is weird is that this mass is about 20 times more than what was expected to be found. Chromosomes are useful in the body because they act as a sort of spool around which DNA is wound. This ensures DNA is packaged efficiently and effectively, and you don't have random parts of DNA being activated or accessed. It's also the cause of certain diseases, particularly those associated with having too few or too many of these chromosomes. Much of the research into those sorts of diseases and conditions is driven by animal models or, in some rare cases, embryonic research. The problem with embryonic research, at least, is that it's nearly always capped at 14 days. This means you have only limited information for the effect it has on a human, either once they're grown or after that 14 days of development has occurred. This is due to a range of issues, primarily those that relate to ethics. Whether or not you can develop an embryo beyond this point, is it right? Is it alive? Is it a human? Many things that are quite thorny issues. In fact, these issues become even more complicated when you start looking at whether or not they're doing chimeric research, that is, half human, half something else. These studies were also held to the same limitation. Scientists now are looking at whether or not that can be pushed out further. In fact, the question is whether or not the 14 days is even necessary. The 14 days is based on 14 days after fertilization, rather than development, and this can mean several things. Notably that if fertilization takes longer, or if there's issues with the time frame for development, the amount of information gleaned can be quite limited. The reason it was accepted is that the International Society for Stem Cell Research, which generally bills itself as being the peak body representing stem cell research, accepted that as a reasonable limitation. The problem is that we're not able to get the information needed anymore from that. In 4 years, we've reached a point where 14 days may not be enough anymore. That peak representative body has said that funding bodies, academics, and academies of science should look at whether or not that is going to be publicly addressed as a concern. That is, engage with the public and get them to understand what sort of research would be extended beyond that 14 days. To start with, we're now able to keep an embryo alive beyond that 14 days outside of a living being. And this meant that before now there was no reason to have to look at extending it. But now we're at that point where we can use the various techniques available to push beyond that and get more information. Information that could be useful in trying to understand and then develop treatments for developmental disorders. The final item of news we have for you this week regards China's artificial sun. This is a fusion reactor and has been an attempt to harness power via a new method. Historically, we've never really had fusion as an option. Fission has worked where we've broken down atomic particles to release their energy, exactly what you see happening in a nuclear power plant. Fusion, on the other hand, is slightly different and much harder to achieve. The Academy of Science in China has managed to get their fusion machine to reach 120 million degrees in Celsius. This is 216 million degrees Fahrenheit if you insist upon using the barbaric imperial system. That temperature was maintained for a little over 100 seconds. To put this in some context, our own sun sits at 15 million degrees Celsius. Of course, that's also been burning at that temperature for millions and millions of years, rather than 100 seconds, so there is something to be said for the sun's efficiency. The key takeaway here is that it's a huge breakthrough and movement above where they were previously. They're able to keep a stable temperature that's incredibly high for a relatively long time. In theory, having a fusion reaction would allow the creation of larger and more complex elements from smaller, more simple ones. Think of what would happen if you took two hydrogen atoms and put them together. Now you suddenly have helium. Helium is an element that we don't have enough of on Earth and we're already running low on. Artificially generating it would mitigate some of the issues where it's used in essential services, such as medicine. Not having any helium reserves means you can't have certain devices used in medical services, things like MRI machines. Being able to create it, although far more expensive and difficult, would allow you to work around these problems to some extent. Of course, such lofty goals as creating more advanced elements from more basic ones is a long ways off. You're hardly going to create anything worthwhile in 100 seconds. In fact, it's likely that we're decades away from achieving anything close to that. That's all the news we have for you this week. Thank you for watching. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.